This is a production of Cornell University.
All right, thank you. So today I'm going to be talking about the role of cucurbitacins in insect preference in Cucurbita pepo. So for those of you who have heard me speak before, you know that, or you may remember that I have been working for the past few years on trying to understand the genetic and biochemical basis of striped cucumber beetle preference in Cucurbita pepo, which is like summer squash, acorn squash, and zucchini. And much of this work has been focused on resistance in leaf tissue, since that is a tissue that matters the most to us here in New York when we transplant squash into the ground. So it's been looking for preference at that level. And much of this work has started with looking at screening germplasm for beetle damage and other beetle-centric bioassays to try to understand the genetic basis of preference and also identify traits that are responsible for beetle preference and do selection for non-preferred varieties. Unfortunately, these beetles are somewhat challenging to work with in the sense that they have seasonally dependent behaviors like mating and aggregation, which can really confound some results and has unfortunately not led to a lot of gain in this regard. So we're trying a few other approaches. So one of the approaches I'm trying is working with a proxy insect for these beetles that neither mate nor aggregate. And I'm using this as a proxy for selection for these beetles, as well as using them to identify other chemical traits in plants that impact insect preference. And then another thing I'm doing, which was what going to be the rest of my talk is about today, is looking at traits in plants that we hypothesize may be important for insect preference, genetically mapping them, creating populations with high or low of these compounds, and then seeing how the beetles respond to these plants. So the last part is what I'm going to be talking about today, and the compound in question in the title of my talk is cucurbitacins. So I'm going to start with an overview of what cucurbitacins are, tell you about some mapping results we have, and then, of course, talk about what this means for the striped cucumber beetle. So cucurbitacins are intensely bitter and toxic triterpenoids. There's more than 20 types of cucurbitacins in the Cucurbitaceae family. And everything we know about their biosynthesis so far is from cucumber, another member of the family. And the general pathway in cucumber is that acetosqualene, an output of the mevalonic acid pathway, is cyclized in the cucurbita dienol by the bitter gene. And then there's a number of other steps of oxidation and to create the final cucurbitacin product. And in cucumber, if plants have the functional bitter allele, there will be cucurbitacins in the cotyledons. And only if the plants have the bitter fruit or bitter leaf transcription factors is the bitter gene expressed in those tissues and cucurbitacins that are in those tissues. And then for how cucurbitacins relate to insects, there's kind of two main classifications of how organisms deal with cucurbitacins. So specialist beetles, like the striped cucumber beetle, have co-evolved with cucurbits, and they actually sequester these cucurbitacins for their own defenses. And so they love these high cucurbitacin, whereas avoid low cucurbitacin plants. On the other hand, other herbivores like spider mites and us, we find high levels toxic, but are fine with lower levels. So I'm going to start with talking about our mapping work in Cucurbita pepo. So Cucurbita pepo has 4 types of cucurbitacins, D, B, I, and E. B is the most abundant, and we measure it with HPLC. It's highest in the cotyledons and low in leaves. So I mentioned earlier that much of my previous work had been in leaf tissue. Everything I'm going to talk about for cucurbitacins is all measuring and selecting in the cotyledons because that's where the compounds are the highest. And in thinking about what germplasm to use for this mapping, it's important to know that in Cucurbita pepo, there are 2 subspecies, pepo, which includes pumpkin and zucchini, and pepo texana, which includes acorn squash and summer squash. While they look quite similar, they're indeed from separate domestication events and quite genetically distinct. And we know from previous work that beetles really prefer pepo, and cucurbitacin cotyledons are high in pepo. And the reverse is true in pepo texana. So in knowing our 2 phenotypes are aligned with population structure, we need to make inter-subspecific crosses and evaluate these phenotypes in those. So that's what we did, is we made a biparental population between a high cotyledon cucurbitacin parent from pepo and a low cotyledon cucurbitacin parent from pepo texana. And the high parent has, again, cucurbitacin B is the highest, D, and then followed by D, E, and I. Expression of all of these are highly correlated. So if you're high in B, you'll be high in the rest of them. And while it's hard to see, cucurbitacins E and I are appreciably higher in the high parent than the low parent. So I looked at 200 F2 individuals. We genotyped them with GBS and phenotyped them for all 4 cucurbitacins. And then we did QTL mapping, and we found one pretty convincing peak on chromosome 5. Because it's an F2, we don't have a ton of recombination in the region, so it's about a 500 kilobase window that we have, but we're working to fine map that further. Interestingly, this is not the bitter gene homolog that we know from cucumber. That's on chromosome 12, and if you zoom in, there's nothing close to significant. So that's one line of evidence that the difference that we see between these 2 populations is not due to the bitter gene. I also developed PCR primers and Sanger sequence this bitter gene homolog, and there were no indications that the protein product would be any different between these 2 parents. So it seems like there's a single gene making these different, and it's not the bitter gene that we know so much about from cucumber. I also, in this F2 population, looked at the highest and lowest producing F2s with 3' RNA-seq to see if there are any genes expressed in this region or elsewhere that would be responsible for this phenotype. And our most differentially expressed gene was indeed in this region on chromosome 5, and it was an excita reductase, which you remember from the pathway for cucurbitacins. There's a series of oxidation steps to make the final product. So it seems plausible, and it is highly expressed in both the high parent and the high F2s, and not expressed in the low parent and low F2s. So yeah, so we think we found possibly a single gene that controls cucurbitacin content in Cucurbita pepo. And then bring this back to what the motivation for this, what the implications for striped cucumber beetles are. So I'll remind you again that there's these 2 subspecies, pepo and texana. If you recall that our high cotyledon cucurbitacin parent was from Cucurbita pepo, beetles love both leaves and cotyledons from this species. So we expect that the high parent's going to have a lot of leaf and cotyledon damage, and the reverse is true for texana. What I haven't told you yet is that leaf cucurbitacins are low in both subspecies. I mentioned they were low, they're low across the board. And even if we try to induce cucurbitacin defenses through herbivory by the striped cucumber beetle or other forms of herbivory, the plant doesn't ever respond by producing more cucurbitacins. So then to answer the question of what selecting for cotyledon cucurbitacins means for striped cucumber beetles, from the F2 population I told you about, I selected the extreme F2s and I selfed them to make F2 derived F3 families, about 15 from either extreme, and I measured damage in the field. So starting with cotyledons, so you can see our high parent gets a lot of, I'm sorry, I'm not sure why that's like that. Well, it's supposed to be 50 and onward. So anyway, so our high parent gets a lot of cotyledon damage, and our low parent doesn't, like we would expect because of the subspecies difference and because of the cucurbitacin difference. And when we select for cotyledon cucurbitacins, we see the same pattern for beetle damage in the high cucurbitacin F3s and low cucurbitacin F3s, which is good. That's what we would expect based on the evolutionary history of the beetle with this compound. And nice to see that this would work as a selection strategy for defending cotyledons against beetles. On the other hand, in leaf tissue, so again, these are these 2 separate subspecies, so we expect the high parent from the preferred subspecies to have a lot of damage compared to the low parent. And we see that again. And there's a few reasons why we may believe that selecting for cotyledon cucurbitacins may affect leaf damage. So for instance, beetles could be attracted to the cotyledons and then move up to the leaves and damage the leaves from there. Or perhaps there's other related terpenoid compounds from the biosynthetic pathway that provide a signal that attracts the beetles to these plants. However, we don't see any difference in leaf damage between these 2 selections. So we find that leaf damage is independent of cotyledon cucurbitacins. So what does control leaf damage? Well, I measured a lot of things, including the phloem sap, how sticky it is, and like the mandibles of beetles, leaf nutrition, and neither of those are appreciably different between the subspecies. However, from some of the work with the insect proxy I was telling you about, we have some exciting results about volatile deterrence in the non-preferred subspecies and are making progress, hopefully, in selecting with this proxy insect. So in summary, we've mapped a cucurbitacin biosynthesis loci in Cucurbita pepo and showed that low cotyledon cucurbitacins reduces beetle damage in cotyledons, but leaf damage is independent of that. And with that, I would like to thank my lab and committee, a lot of people in the chemical ecology area, and all of my funding. Right, thank you. Does anyone have any questions?
I guess this is just a question for me, methods-wise. When you make a mapping population, you have two. How do you phenotype? Is it just like you got eaten by a beetle and you didn't? Is it just damage phenotyping?
Yeah, so for this, we phenotyped the cotyledon for cucurbitacin content. So we just cut off a cotyledon and extracted from there, and then took DNA from the leaf tissue later. Okay. But in previous mapping work, we have, just looked at damage to leaves and then sequenced them later. Yes?
So the different classes of insect predators, like the specialized versus the more general ones, if you reduce the cucurbitacin content, you're going to get more general ones. So is there like a balance, or is New York have a specific balance that you're trying to go for?
That's a great question. So the question was, if you are selecting for lower cucurbitacins to protect against beetles, that can be a problem for other insects. And the answer is yes, it very well could be. But kind of like you were alluding to, actually, in New York, the striped cucumber beetle is by far our biggest pest problem. And it's, so it's kind of, and we don't have a lot of other pests that we see as important as a striped cucumber beetle. So it'd be worth a trade-off in this region. Yeah.
So just to clarify for myself, the hypothesis is that these volatiles potentially, are sort of correlated in the parents. So cucurbitacins goes with certain volatiles in the one parent and not in the other parent. But then once you make the mapping population, they're uncorrelated in the mapping population.
Yeah. So I probably didn't do a great job explaining this. So the cucurbitacins, they're pretty large compounds, so they are not volatile, but other parts, like intermediate steps, are small enough that they could be volatile compounds. And our hypothesis for why high cucurbitacin content in the leaves could be due to some of those intermediate or byproduct compounds. But the volatile result I was talking about has nothing to do with cucurbitacins. We think it's we don't know what it is, but it seems to be a separate compound.
So sort of following up on that, are you going to do any GC to look for other volatiles?
Yes. Yeah, I don't know anything else. Yeah. Great. All right. Thanks, guys.
This has been a production of Cornell University, on the web at cornell.edu.